Hello Year 10, I hope you're all staying well and safe at home and getting on and with all of your work. So today's lesson is a recap lesson from Year 9, like all the other lessons in this unit, and it's called Discovery and Development of Drugs. Now what I will say is that throughout the video I would ask you to pause the video and answer some questions or complete an activity, and you're more than welcome to take notes as I go through the video. Just pause me and take some notes as you feel free. So our objectives today are: I can describe how penicillin was discovered, I can describe how scientists test and trial drugs, and I can explain why it is important to test new drugs. So we'll start off with the discovery of drugs, and then we'll move on to the development of drugs afterwards. So as always, here's your do it now task. The 3 multiple choice questions. So I suggest you pause the video, gives you time then to write down the date and the title, underlined in pencil of course, and then attempt the 3 tasks. So pause the video now please. Okay, let's go through the answers then. So number 1, who discovered penicillin? Hopefully you remembered it was Alexander Fleming, so tick it if you got that one right. And which tree bark did people used to chew on to relieve pain? So you know what bark is, it's that brown stuff that goes around the trunk of a tree. People used to chew on that because of its pain relieving qualities, and of course that was the willow tree. And a non-communicable disease definitely cannot be passed on to someone. So well done if you got all 3 right. Right, let's move on then. So some interesting facts before we start. I was quite interested in this when I was researching this topic. So the first one, there's this chemical called digoxin inside the foxglove plant, and then they are on the right hand side. In the 18th century, it's about 300 years ago or so, they were administering that to people to help strengthen their heartbeat, and that's still being used today. Brilliant. This one's quite gross. There must be something in bread that the ancient Egyptians knew about thousands of years ago. So if you got a cut and it went septic, they would let bread go mouldy and they would rub that all over the septic wound to try and clean it up. And the last one's quite yucky as well. So of course in the willow tree, the bark contains these pain relieving chemicals, and the beaver, like the animal down in the bottom right, eats this bark, and all those chemicals actually concentrate in their tail. So a long time ago people would chew and suck on beaver tail, and then they would get those pain relieving chemicals into themselves. Even if I was in pain, I'm not sure I could do that to a beaver. I don't know. What about you? Could you do that? I'm not sure. Right then, let's move on to our actual sciencey lesson then. So in 1928, there was this really famous scientist we talked about a minute ago called Alexander Fleming. And what he was doing was growing all sorts of bacteria in petri dishes to see what would happen. And there's one incident that he left the lid of the petri dish off, and then he went on holiday. Now you've got to imagine, if we did that today, you're growing all sorts of bacteria, all sorts of weird bacteria, and you're growing them on agar or some kind of nutritional jelly. You leave the lid off and then off you go. You go on holiday somewhere. I mean, health and safety would have a field day with that. It just wouldn't be allowed. You can't just let bacteria grow in all sorts of places. That's just ridiculous. But he did, and when he came back from holiday, he realised that the mould had grown in the middle as expected, and bacteria had grown all over the jelly as expected. But what he didn't expect was this ring around the mould, and this ring didn't have any bacteria on it. In fact, nothing grew on it. So what Fleming had realised was that there was something in the mould that killed all of the bacteria. And of course, you'll know from your Year 10 lessons, this is now called the zone of inhibition, and you can see that all the bacteria is killed. So there must be something in the mould that's killed that. And because of the name of the mould, Fleming called it penicillin. So here's another picture of Alexander Fleming on the right, looking very, very proud of his achievements. Some people in the world are allergic to penicillin. You may be yourself. But for those people who are not, penicillin has helped millions and millions of people ever since he found it. And on the left-hand side, you can see somebody else has had a go at doing this and got exactly the same results. But instead of one mould, they've got 7, and you can see the rings around the mould where no bacteria has grown. Now, Fleming knew about penicillin, but he couldn't actually get the penicillin out of the mould itself. And we had to wait nearly a decade, so 1938. Let me move my head out of the way. for two scientists called Ernest Chain and Howard Florey, and they managed to get the penicillin out of this kind of like mold juice. It sounds a bit disgusting, but that's where it was. And what they decided to do was to see how effective it was. How effective was this penicillin? They knew it killed bacteria on the petri dish, but they wanted to test it on a live human being. So they found a man in a hospital who was dying because he had a blood infection, and they gave him the penicillin, and he got much better. But they didn't have enough penicillin, so of course the blood became infected again, and eventually the man passed away. Chain and Florey were not put down by this. They went and made lots and lots more. In fact, they went into industrial scale manufacture of penicillin. And of course, you'll know from your history lessons that 1939 was the outbreak of World War II. And penicillin was made on industrial scale to help all the wounded soldiers in World War II. So you can imagine how many people might have died in World War II had it not been for Florey and Chain from extracting the penicillin. Okay, so times have moved on. We can't just go around putting new drugs that we think might or might not work into people in hospitals. I mean, that's just crazy to think about. So we have these people who do this job called research chemists, and they use things like chemical banks and computers to manufacture or synthesize new drugs. And in Costa Rica, people have been drinking this fruit juice from a fruit called noni, and you can see the noni on the left hand side. To me it looks a bit like a lime potato, mixed with a green lime and a potato, but it's the juice itself that can treat infections and non-communicable diseases like diabetes and high cholesterol. And you think about it, over in this part of the world we don't know what this fruit is. Why aren't we all drinking this to stop us getting diabetes and problems with our cholesterol? I know in Costa Rica they do. So these research scientists are trying to find a way for that noni fruit to be used in a pill form that we could take as a natural antibiotic. Or maybe there's a way that they can synthesize, they can manufacture in a lab the juice from a noni fruit. And there's an advantage to that. We don't want to grow lots and lots of noni fruit and then have to transport them and everything. If we can just make it in a lab anywhere in the world, then that would save a lot of time and a lot of money. So some bacteria can actually develop and it becomes resistant to antibiotics. So you may have been ill and a doctor might have prescribed you antibiotics. For some bacteria, those antibiotics won't work on them. And when this happens, the antibiotics don't kill the bacteria. So looking in soil, scientists, they find the microorganisms inside the soil, and they try to culture them, because if they do that, then they might be able to find some antibiotics in there. And when they look at soil, they used to only be able to look at about 1% of all the soil. So 99% of the soil would be never looked at. And then somebody invented this machine, little tiny contraption called the eye chip. And it's full of little pockets, and the bacteria from the soil or from the sea or wherever gets trapped in those little pockets. And inside each of those little holes, you can see in the picture there, it's full of agar jelly, and this is like food for the bacteria. So instead of only looking at 1% of the soil, now they're able to look at up to 99% of the soil. So it's made their job 99 times more efficient. They're able to sift through 99 times more soil. In 2015, something absolutely incredible happened using this little tiny eye chip. They found this thing called a super antibiotic. So they'd found one of these little bacteria produced this super antibiotic, and when they tested it, they found out that it killed things like MRSA, tuberculosis, and all these kind of bacteria that were resistant to antibiotics. It wasn't resistant to this, and this was able to kill that bacteria. In fact, it kills practically everything except for E. coli. It's worked on the mice, but it hasn't worked on human beings yet because they haven't tested it yet. It was only done in 2015, so not that long ago. But it wouldn't surprise me if in the near future we started seeing these super antibiotics prescribed to us by doctors. So, using this technology, like we talked about with the noni fruit, scientists were able to extract just the important chemicals or the important compounds from them. So if you've got a microorganism or a plant, they can just extract just the little bit that they want. and then they're able to make it in the lab over and over and over again, or they can modify it, or they can add things to it to make these molecules even more powerful. And this is really good because it means that they can synthesize or make these drugs much more easily and much more cheaply. It means, like the noni fruit, like I said earlier, you don't need to have all that fruit transported all around the world. They can just make it using chemicals in the lab, and they make exactly the same chemicals as what's found naturally in the fruit. So this is your opportunity to have a go at three questions. What I want you to do is to time yourself 10 minutes. Just give yourself 10 minutes. You don't need to go any further than that. And I want you all to start with amber. Let me move my big head out of the way there. So all start with amber, move on to green, and then see if you can complete the Be Awesome task as well. So 3 marks for the amber. Describe how Alexander Fleming discovered penicillin. 3 marks for green and 6 marks for the Be Awesome task. Okay, so I suggest you pause the video now and time yourself 10 minutes. So off you go. Okay, so we're going to move on. You'll get your answers for this next lesson. So hopefully you've done this and then you're able to send a copy to your teacher. So now we've looked at the discovery of drugs and how penicillin was discovered and how it was able to be extracted and how it is used. Now we need to look at how drugs are developed. You can't just do what Jane did and just gave it to another human being without any testing. Who knew what the side effects could be? I think they were just lucky with the penicillin. So scientists can develop new drugs in a lab, and laboratories, it just means lab, doesn't it? And all new drugs need to be rigorously tested and trialled before they go to market. In fact, now we test and trial those drugs much more than we used to in the 1970s or 80s. So there's a picture of somebody doing just that. And after trialling and testing, drugs have to be these 4 things. They have to be effective, so they've got to prevent or cure a disease so that it can make you feel better. They've got to be safe, so they can't be toxic. Toxic is just another word for poisonous. You don't want it to be poisonous inside you or have any side effects that are not good, So it has to be safe. It's got to be stable. You don't want to have it above your fridge or something, or in a box and it all breaks down. So when you open up the packet, there's nothing in there, or it turns into something else. You can't have that. So it's got to be used in normal situations and be stored. And when it goes into your body, it needs to be able to get out of your body. It needs to be able to clear itself from your body as well. These 4 statements, effective, safe, stable, and successfully taken into and removed from your body, are really, really important. And I suggest that you pause the video and copy these into your notes. So pause the video now and do that, please. All right, let's move on. So develop and market an actual drug can take up to about 12 years and cost almost £2 billion. That's a lot of money. So researchers will look at a disease that exists and they'll make a whole load of drugs that they think will work at that disease. But of course you can't just go and send out into the public all these different drugs. They have to look at the toxicity, that's the poisonous, how poisonous it is, and whether or not the drug actually does the job it's supposed to do. So in the lab, they'll test that drug on things like cells, like a skin cell, and maybe the tissue, so maybe lots of skin or some muscles. And then they'll test it on some organs. They might test it on the liver, or they might test it on kidneys, something like that. And not many drugs actually get past this point. At this point, most drugs have found to be too toxic or poisonous, or they just don't work very well. So it's kind of like back to the drawing board again, and let's change that drug. Let's see if we can make it do its job. So here's a graph here on the right hand side. And what does the graph show? So have a little look at the graph and write down the answer in your book, please. So pause the video and write the answer to what does this graph show. Okay, so the graph shows preclinical and clinical trials for each decade from the 1970s up to 2010. And you can just see that the cost of preclinical and clinical trials has gone up and up and up and up and up. So we are much better today at testing all these drugs, making sure that nothing can go wrong than we ever used to be. So if the drug does get past that point, so it's not toxic and it does exactly what we want it to do, then we have to do some animal testing. So the drug has to be tested on animals. And a lot of people are anti this, and I understand why. I can see why people don't like the idea of drugs being tested on animals. But to play devil's advocate and play it in the other hand, would you rather test a drug on a human being or a child or even a baby, or would you test it on a rat or a monkey? And there's lots of ethics to consider with this. PETA, or PETA, they don't like the idea of animal testing. You've probably heard of this organisation. And they've strived forward in reducing the amount of drugs that are tested on rats and monkeys. So what we need to do, or what scientists need to do, because I don't do this, is see how the drug responds to these animals and see if there are any side effects. As I said before, you wouldn't just go and test on a human because the side effects could be disastrous. So they test it on the animals to see if there are any side effects. And then by testing on animals, scientists can kind of predict how it's going to affect the human being. And up to this point, seeing if the drugs are toxic, seeing the side effects and animal testing, all of this is called preclinical testing. Okay, so once they've done that, they've tested it on the animals and it's passed, there hasn't been any side effects on the animal. Now we move on to something called clinical trials. Now, this is where it does start to be tested on human beings, and people actually volunteer for this. They volunteer to have a brand new drug administered to them to see what the side effects are. I personally would never do this, but I know that I've known lots of people who have done this. So people volunteer, and then they give them blood tests and all sorts of things and questionnaires to make sure that they are healthy. Even scientists do this. They don't want to administer a drug into somebody who's poorly or who has a disease. Have to put it into healthy people. But the dose of the drug that they put into them is very, very low. Very, very low dose, just in case that there might be a side effect. And they don't want them to have a side effect, obviously. So if that's successful, so they've done very low doses on a small amount of people, on these volunteers, and they're all okay, then they find people who have the disease that this drug is supposed to help prevent or to try and combat. And then they test it on these people and see how it fights the disease. So the drug seems safe. They've tested it on the people with the disease and it seems safe. Then they do a bigger clinical trial. And what they do, they up the dose each time on these people to see what the optimum or best dose is. So originally they started off very low, then they tested it on people with the disease with a low dose, and then they up the dose to see what the best dose would be for the drug. And then if it passes all of that and everybody's fine and it's not toxic and there are no side effects, then doctors would be able to prescribe it to you. Okay, so let's have a look at what goes on in these clinical trials. So imagine you've got a group of people and they've got a target disease and this drug is supposed to combat that disease. So what scientists do, they randomly give some of these people a placebo. A placebo is something that doesn't contain the drug that they are testing. And then they give other people the new medicine. So there's a picture there of placebo and the test drug. So this way, the doctor doesn't know who's got placebo, who's got test drug. It's completely anonymous. The people themselves don't know if they're taking the placebo or the test drug, because there might be some sort of cognitive bias. If the doctors knew, they might try to accidentally, without knowing, might doctor the results. There might be some bias. So it's just best that nobody knows and it's completely anonymous who takes what. Now, the placebo back in the past used to be a sugar-coated pill. And if somebody actually had a disease and this drug was supposed to help them combat that disease, the last thing they would want is a sugar-coated pill, because that's not going to help them with their disease. So more recently, what they do, the placebo isn't a sugar-coated pill. They give them another form of medicine that's going to help combat the disease. So in the end, they're still going to be fixed. They're still going to feel better because they know the drug works. But what scientists are able to do is determine if the new drug works as good as what the placebo does. So the whole idea of placebo has changed over time. I mean, can you imagine? You go in for drugs trials and you've got a disease and then they give you a sugar pill and they think that that's going to work. Of course, it's not going to, is it? You're still going to be ill. So once they've done that, both sets of patients are compared, and then the doctors then will write their report. Now, it's all well and good that the doctors have all this data and they can say, yes, look, my drug works with my group of people. Other scientists around the world, they need to replicate this. They need to copy the same trials and the same testings on different groups of people to see if it works for them as well. And then if it does work, then the drug and all the information and all the research can be published in a scientific journal. And it's here where it can be peer reviewed. Peer reviewed means when other scientists have replicated what you've done and then they've got the same answer. That's the whole idea. Sometimes it doesn't come out with the same answer, but when it does, it can be published. And there's this organisation called NICE, which is NICE, National Institute for Health and Care Excellence. They then, if they like the drug and they think there's a lot of use for it, then they can recommend doctors in the NHS to prescribe it. So that's our development of drugs bit done. And just like we did at the beginning, halfway through the discovery of drugs, and give you a time limit. Let's start with the amber task and see how far you can get. So this time I want you to give yourselves 12 minutes. So there's amber, there's green, and there's the be awesome task. Again, if I move my big head out of the way, you can have a go at this. So time yourself 12 minutes and answer the 3 questions. Okay, off you go. Pause me, please. Okay, so as before, we're not going to give you, I'm not going to give you the answers just yet. You're going to have to wait till next lesson to be able to mark your work. I suggest you take a picture of your work and then you upload it to Google Classroom straight away so that you don't forget it, and then your teacher knows that you've done the work. Okay, I hope you enjoyed discovery and development of drugs, and I'll see you next time. Take care and stay safe, everybody. See you soon.